They have no leaves, no roots, and no sunlight, yet they live everywhere, hidden in the soil, in your bread, even on your skin. Welcome to the kingdom of fungi. And if you think you know what fungi look like, get ready to be surprised. Basidiomycetes, from the ancient Greek basidion, base, are probably the fungi that first come to your mind. Nothing unusual so far. Porcini, fly agarics, russulas, ascomycetes, from the ancient Greek word ascus, meaning a small sac, are rather strange fungi. The morel still looks somewhat like a mushroom, but the one next to it, excuse me, what is that?
Chytridiomycetes.
What are these and where are the fungi? Oh, do we need a magnifying glass for that? What's this? Some kind of mold? Oh, that's right. These are the Mucormycota, named after the Latin word mucor, which means mold. But that's not a fungus. It's a fly. Ew, its belly is swollen and white. Oh, that's because it's been attacked by predatory Zoopagamycota fungi, from the Greek words zoo, meaning animal, and pagos, meaning attached. And here, a mask. Why? Because in the past, Deuteromycetes was the name for all fungi whose sexual stage couldn't be found. Such secretive creatures. But scientists decided to stop spying on the fungi's private lives and instead determine their relationships by DNA. That turned out to be much easier, and almost all Deuteromycetes move straight into the ascomycetes. Differences between major groups of fungi lie in how their spores are formed, and to understand this, a microscope is essential. Let's start with the familiar Basidiomycete fungi. They form the fruiting bodies you know well, mushrooms with a stalk and a cap. Most of the time, the fungus lives hidden underground as a network of hyphae. But spores can't spread effectively from there, so sometimes the fungus lifts up, forming a visible fruiting body. The spores are located on the underside of the cap, usually in a tubular or lamellar hymenophore. Under the microscope, each segment of the hymenophore reveals a dense layer of basidia, specialized hyphae that produce spores on their tips. The name, Basidiomycota, comes from these structures.
Each basidium contains 2 haploid nuclei. To form basidiospores, these nuclei first fuse into a diploid nucleus. Then meiosis occurs, producing 4 haploid nuclei. Each nucleus migrates into a small projection on the surface of the basidium, where a basidiospore begins to form. When mature, the spores are released into the air. Each basidiospore can germinate into a haploid mycelium containing 1 nucleus per cell. For a new mushroom to grow, 2 compatible mycelia must meet and fuse, forming a dikaryotic mycelium with 2 nuclei in each cell. This mycelium is capable of producing new fruiting bodies and new basidiospores, completing the life cycle of basidiomycete fungi.
Ascomycetes produce ascospores. Ascospores develop inside asci, sac-like structures that form at the tips of ascogenous hyphae. The asci are grouped into fruiting bodies called ascocarps. If an ascocarp is completely closed and spherical, like in powdery mildew fungi, it is called a Cleistothecium. If it resembles a flask with a narrow opening, it is a perithecium. And if it is open and shaped like a small saucer, it is an apothecium. Ascospores are released from their ascus and germinate into haploid hyphae. Then, on one hypha, an ascogonium develops, and on another, an antheridium. In both structures, the nuclei divide metodically, producing several nuclei in each cell. a special outgrowth called a trichogene appears on the ascogonium, forming a bridge between the ascogonium and the antheridium. Through this bridge, nuclei from the antheridium move into the ascogonium. After the nuclei are transferred into the ascogonium, the main process begins. Ascogenous hyphae start to grow from it. There can be many of them, but for simplicity, only one is usually shown. In an ascogenous hypha, the nuclei are arranged in pairs, N plus N. The tip bends to form a hook. At this moment, the nuclei near the tip divide mitotically. The two uppermost nuclei fuse, forming a diploid nucleus. Cross walls, septa, appear, and the upper cell becomes an ascus. Inside the ascus, the diploid nucleus undergoes meiosis, producing 4 haploid nuclei. This is followed by another mitotic division, resulting in 8 ascospores enclosed within the ascus. I think you've already guessed what the spores of chytridiomycetes are called. That's right, zoospores. Zoospores are not uncommon in the animal world, but we're dealing with fungi here. And for them, this is a unique and taxonomically important feature. Chytridiomycetes don't have complex spore-forming structures like ascomycetes or basidiomycetes. Their zoospores are their main distinguishing feature. These are motile spores with a single flagellum that allows them to swim in water. No other fungi, at any stage of their life cycle, have motile cells. Now you're probably wondering, how do the spores of Mucoromycota and Zopagomycota differ? Sorry for not making your life any easier, but almost not at all. Both form zygospores. By the way, zygospores are also found in some algae, so it's not really a fungal invention. Once again, we'll need a microscope to see how zygospores are formed. During zygamy, 2 hyphae of opposite mating types grow toward each other and fuse at their tips. At the point of fusion, their cytoplasm merges, forming a young zygospore. Inside it, many haploid nuclei from both hyphae are initially present. However, only 2 of them, one from each parent, fuse to form a diploid nucleus. The remaining nuclei gradually break down. The zygospore develops a thick wall and enters a resting stage. It can remain dormant for several months, or even years, waiting for the right conditions to germinate. When those conditions arrive, the diploid nucleus undergoes meiosis, and the zygospore germinates, beginning a new life cycle. Now you're probably thinking, finally, we've figured out all the spores. If Deuteromycetes, that is anamorphic fungi, have no sexual reproduction, then maybe they don't have spores at all. But they do. In anamorphic fungi, spores form asexually. Their spore-bearing structures can be surprisingly complex and diverse. You could almost make an ikebana out of them. That's exactly how scientists tell them apart under the microscope. Look at the beautiful spore-bearing structures formed by Penicillium, Trichophyton, and Candida.
Great! We've figured out what's so special about fungal spores that scientists separate fungi into different phyla. To complete the picture, we just need to look at a few representatives from each group.
Basidiomycetes include chanterelles, delicious mushrooms that look just as bright and golden in soup as they do in the forest. Tremella looks like a piece of clear jelly that somehow grew on tree bark, though in fact, it's a parasite of another fungus. If you really want to, you can eat it. What are those spots on the leaves? That's a microscopic member of the Basidiomycota, hollyhock rust. Looks tasty? You'd be right to think so. This is corn smut. Despite its rather unappetizing appearance, it's perfectly edible and even considered a delicacy in Mexico. The lingonberry exobasidium turns the leaves of lingonberries, azaleas, or camellias bright red and fancy. Beautiful, perhaps, but not for the plants. They can actually die from it. Oh, there's an ear growing on the tree. It's a fungus called the jelly ear. In Chinese cuisine, it's considered a delicacy. The picture is already looking cheerful, but now we're moving on to the ascomycetes. And things are about to get even more colorful. The false morel looks like a true morel that got slightly flattened with a fly swatter. Don't mix them up. The false morel is poisonous. Baker's yeast helps make delicious bread and pastries. If your bread is covered with gray-green mold, it's most likely penicillium. Claviceps purpurea is the fungus that causes rye ergot and produces toxic alkaloids. If people eat the hardened sclerotia, mistaking them for grains, they can experience hallucinations, convulsions, or even gangrene. This condition is known as St. Anthony's fire. It's believed that ergot poisoning may have been behind some of the mass hysteria and possessions of the Middle Ages. Epidermophyton feeds on keratin, the substance that makes up our skin, hair, and nails. You might have seen its traces on someone's feet at the pool. The skin turns red and flaky, and the nails become thick and yellow. It's yaks, and highly contagious.
Neolecta looks like a yellow or orange stick about the height of a hand. You'll most likely find it under a spruce tree. It loves the symbiotic fungi of conifers and lives among them as a saprotroph. Now let's add some Chytridiomycota to our picture. Opidium brassicae causes black leg disease in cabbage. Oh no, my cabbages!
And this potato is home to Syncytrium endobioticum, the cause of potato wart disease. Definitely not something you'd want to eat.
And this strange little monster is Rhizophydium globosum. Its body has two parts, a branching network of rhizoids that absorb nutrients and a sporangium that produces zoospores.
This is a larva of a malaria mosquito. It didn't survive its encounter with the fungus Colomomyces stegomeae. That's why this fungus can be used as a biological control agent against yellow fever carriers. A cow is certainly not a fungus, but in its stomach live many symbionts that help digest cellulose, including the fungus Neocalamastix. Now let's see who we have here among the Mucoromycota. Look at this curious little thing growing on the sphagnum moss, and it even smells awful. No way we can just walk past that. Think the rodents as they munch on the bright sporocarp of the fungus endogone and spread its spores around with their droppings. Pilobolus grows on dung and shoots its spores so accurately it can hit the next pile of poop. The fungus mucor grows very fast, and in no time it spoiled this strawberry. You can eat it if you really feel sorry for the berry, but it's neither tasty nor good for your health.
And who do we see among the Zoopagomycota? The example in the picture is the fungus Entomophthora muscae. It infects houseflies and can cause epizootics, large outbreaks among insects. Well, that's nice. That's good. Those flies were getting on everyone's nerves anyway.
The fungus Piptocephalis is also a predator, but it prefers cannibalism. It feeds on other fungi.
And this is Stylopage. Want to hear how it hunts? Its future victims, an amoeba or a nematode, touch the hypha and instantly get stuck to the fungus with a drop of sticky substance released at the contact point. Then a haustorium grows from the fungus, penetrates the victim's cell membrane, branches inside, and slowly drains its insides. Once the poor thing is completely emptied, Stylopage discards the useless haustorium and waits for the next victim. Meanwhile, a little pile of empty shells keeps growing around it.
Our story of fungi comes to an end. You made it through, my clever friend.